Hello and welcome, dear students. Today we are going to discuss about the topic mechanism for dynamics in gene frequency. The main objectives of today's lecture are to know the basics of gene frequency, to know the theory of gene frequency, to understand the estimation of gene frequency, to understand the dynamics in gene frequency, to understand the mechanism for dynamics in gene frequency, and to understand about natural selection as a process of genetic change. Dear students, let's start with the introduction first. Gene frequency or allele frequency refers to the proportion of different alleles of a gene in a random mating population expressed as a fraction or percentage. In other words, the proportion of each type of allele at a particular locus in a random mating population is referred to as gene frequency. The composition of a population is described in terms of gene frequencies. Specifically, it is the fraction of all chromosomes in the population that carry that allele over the total population or sample size. Microevolution is the change in allele frequencies that occurs over time within a population. The allele frequency is distinct from the genotype frequency in that although they are related and allele frequencies can be calculated from genotype frequencies. In population genetics, allele frequencies are used to describe the amount of variation at a particular locus or across multiple loci. When considering the ensemble of allele frequencies for many distinct loci, their distribution is called the allele frequency spectrum. In a population, allele frequencies are a reflection of genetic diversity. Change in allele frequencies over time can indicate that genetic drift is occurring or that new mutations have been introduced into the population. Dear students, let's understand the theory of gene frequency or allele frequency first. The theory of population genetics is a theory of allele frequencies. Each gene in the genome exists in different allelic states, and if we focus on a particular gene, a diploid individual is either a homozygote or a heterozygote. Within a population of individuals, we can calculate the frequencies of the different types of homozygotes and heterozygotes of a gene, and from these frequencies we can estimate the frequency of each of the genes or alleles. These calculations are the foundation of population genetics theory. Dear students, let's know about the estimation of gene frequencies. The estimation of gene frequencies in a population consists of three important steps. Number one is sampling. First, a random sample of individual is drawn from the random mating population. The size of sample should be adequate to represent all the individuals of a particular population. Number two is the classification. After sampling, the individuals are grouped into different classes for a gene and their number is counted. Number third is the calculation of gene frequency. Allele frequency or gene frequency is calculated by dividing the number of times the allele of interest is observed in a population by the total number of copies of all the alleles at that particular genetic locus in the population. Allele frequencies can be represented as a decimal, a percentage, or a fraction. Dear students, let's now discuss the dynamics of gene frequency. Population genetics describes the genetic composition of a population, including allele frequencies and how allele frequencies are expected to change over the time. The Hardy-Weinberg law, which describes the expected equilibrium genotype frequencies in a diploid population after random mating. Random mating alone does not change allele frequencies, and the Hardy-Weinberg equilibrium assumes an infinite population size and a selectively neutral locus. In natural population, natural selection, gene flow, and mutation play an important role to change allele frequencies across generations. Genetic drift causes change in allele frequency from random sampling due to offspring number variance in a finite population size, with small populations experiencing large per generation fluctuations in frequency than large populations itself. There is also a theory that second adaptation mechanism exists niche construction. An allele at a particular locus may also confer some fitness effect for an individual carrying that allele on which natural selection acts. Beneficial alleles tend to increase in frequency while deleterious alleles tend to decrease in frequency. Even when an allele is selectively neutral, selection acting on nearby genes may also change its allele frequency through hitchhiking or background selection. While heterozygosity at a given locus decreases over time as alleles become fixed or lost in the population, variation is maintained in the population through new mutations and gene flow due to migration between populations. Because population changes require change in gene frequencies, it is important to understand how these frequencies can change. The three primary methods of change are mutation, migration, and selection. So these three things, that is mutation, migration, and selection, play an important role in change of gene frequencies. Dear students, let's now discuss the mechanism for dynamics in gene frequency change. Mutation, genetic drift, gene flow, and natural selection are the mechanisms that cause change in allele frequencies over time. Mutation, that is the allelic variation that makes evolution possible, are generated by the process of mutation, but new mutations change gene frequencies very slowly because mutation rates are low. A mutation is a change or alteration happening in DNA, gene, or chromosome due to intrinsic or extrinsic factors such as an error in replication or exposure to UV lights, respectively. Change in nucleotide sequence of DNA is called a mutation. Change in nucleotide sequence may be an insertion, deletion, or substitution of a nitrogenous base. Mutation is an important biological process in nature. They account for the variation we see in human hair color, skin color, height, shape, behavior, and susceptibility to diseases. Mutations result in the formation of new alleles. It can be helpful or harmful. For instance, the mutation creates variation in nature by providing new alleles in nature and hence helps in the evolution. We can say that mutations serve as a raw material for the evolution to act. In any case, allelic frequencies usually are not in mutational equilibrium because some alleles are favored over others by natural selection. The equilibrium frequencies are then decided by the interaction between mutations and selection, with selection usually having the greater consequence. Mutations therefore add new alleles into a gene pool. This causes a change in the frequency of certain allele combinations in the population, which will cause the population to evolve over time. They are a major evolutionary force that creates new gene variations. Dear students, let's now discuss the gene flow. Gene flow or gene migration takes place when individuals migrate from one population to another and interbreed with its members. Gene frequencies are not changed for the species as a whole, but they change locally whenever different populations have different allele frequencies. In general, greater the difference in allele frequencies between the residents and the migrant individuals, and the larger the number of migrants. The greater effect the migrants have in changing the genetic constitution of the resident population. Gene flow is a unifying force that prevents populations from diverging. Gene flow thus introduces new alleles that can displace old ones if they are better adapted to the current host. Migration may increase the frequency of alleles in a gene pool by adding alleles, that is the immigration of individuals, or decrease the frequency of alleles by removing alleles, that is the immigration of individuals. Migrants change the distribution of genetic diversity among populations by modifying allele frequencies, that is the proportion of members carrying a particular variant of a gene. The genetic variation found within populations involves the frequency of particular alleles within that population, that is the genetic variation. Gene flow is a major cause of the change of gene frequencies within a population, which has played a role in evolution itself. This new variation can create the overall result of increased fitness of the population to the current or future environment. Gene flow is a fundamental agent of evolution based on the dispersal of genes between populations of a species. It involves the active or passive movement of individual plants, animals, gametes, or seeds. A gene pool is what describes the alleles for a particular trait in a population, as opposed to genotype, which describes the alleles for a particular trait within an individual. Dear students, let's now discuss the genetic drift. Genetic drift, as we say, gene frequencies can change from one generation to another by a process of pure chance known as genetic drift. This occurs because the number of individuals in any population is finite, and thus the frequency of a gene may change in the following generation by accidents of sampling, just as it is possible to get more or fewer than 50 heads in 100 throws of a coin simply by chance. The magnitude of the gene frequency change due to genetic drift is inversely related to the size of the population. The larger the number of reproducing individuals, the smaller the effects of genetic drift. This inverse relationship between sample size and magnitude of sampling errors can be illustrated by referring again to tossing a coin. When a penny is tossed twice, two heads are not surprising, but it will be surprising and suspicious if 20 tosses are yield all heads. The proportion of heads obtained in a series of throws approaches closer to 0.5 as the number of throws grows larger. The relationship is the same in populations, although the important values here is not the actual number of individuals in the population, but the effective population size. This is the number of individuals that produce offsprings, because only reproducing individuals transmit their genes to the following generation or to the next generation. It is not unusual in plants as well as animals for some individuals to have large number of progeny while others have none. In marine seals, antelopes, baboons, and many other mammals, for example, a dominant male may keep a large harem of females at the expense of many other males who can find no mates. It often happens that the effective population size is substantially smaller than the number of individuals in any one generation. The effects of genetic drift in changing gene frequencies from one generation to the next are quite small in most natural populations, which generally consist of thousands of reproducing individuals. The effects over many generations are more important. Indeed, in the absence of other process of change such as natural selection and mutations, populations would eventually become fixed, having one allele at each locus after the gradual elimination of all others. More important, natural selection and other processes change gene frequencies in ways not governed by pure chance, so that no allele has an opportunity to become fixed as a consequence of genetic drift alone. Genetic drift can have important evolutionary consequences when a new population becomes established by only a few individuals, a phenomenon known as the founder principle. The founder principle is one reason that species in neighboring islands, such as those in the Hawaiian archipelago, are often more heterogeneous than species in comparable continental areas adjacent to one another. Climatic or other conditions, if unfavorable, may on occasion drastically reduce the number of individuals in a population and even threaten it with extinction. Primitive human populations of the past were subdivided into many small tribes that were time and again decimated by disease, war, and other disasters. So differences among current human population in the allele frequencies of many genes, such as those determining the ABO blood group and other blood groups, may have arisen at least in part as a consequence of the bottlenecks in ancestral populations. Persistent population bottlenecks may reduce the overall genetic variation so greatly as to alter future evolution and endanger the survival of the species. A well-authenticated case is that of the cheetah, where no allelic variation whatsoever has been found among the many scores of gene loci studied. Dear students, let's talk about the natural selection as a process of genetic change. Natural selection promotes the adaptation of organisms to the environments in which they live. Any hereditary variant that improves the ability to survive and reproduce in an environment will increase in frequency over the generations, precisely because the organisms carrying such a variant will leave more descendants than those lacking it. So hereditary variants, favorable or not to the organism, arise by mutations. Unfavorable ones are eventually eliminated by natural selection process. Their carriers leave no descendants or leave fewer than those carrying alternative variants. Favorable mutations accumulate over the generations. The process continues indefinitely because the environments that organisms inhabit are forever changing. So environments change physical in their climate, configuration, and so on, but also biologically because the predators, parasites, competitors, and food sources within which an organism interacts are themselves evolving. Dear students, let's now have the general overview of dynamics of gene frequency or allele frequency. So mutation, gene flow, and genetic drift are random processes with respect to adaptation. They change gene frequencies without regard for the consequences that such change may have in the ability of the organisms to survive and reproduce. If these were the only processes of evolutionary change, the organization of living things would gradually disintegrate. The effects of such processes alone would be analogues to those of a mechanic who changed parts in an automobile engine at random with no regard for the role of the parts in the engine. So natural selection keeps the disorganizing effects of mutation and other processes in check because it multiplies beneficial mutations and eliminates the harmful ones. Natural selection accounts not only for the preservation and improvement of the organization of living beings, but also for their diversity. In different localities or in different circumstances, natural selection favors different traits, precisely those that make the organisms well adapted to their particular circumstances and ways of life. Dear students, let's now discuss about the gene frequency dependent selection. The fitness of genotypes can change when the environmental conditions change. White fur may be protective to a bear living on the Arctic snows, but not to the bear who is living in a Russian forest. There, an allele coding for brown pigmentation may be favored over one that codes for white fur. The environment of an organism includes not only the climate and other physical features, but also the organisms of the same or different species with which it is associated. So, changes in genotypic fitness are associated with the density of the organisms present. Insects and other short-lived organisms experience enormous yearly oscillations in density. Some genotypes may possess high fitness in the spring when the population is rapidly expanding because such genotypes yield more prolific individuals. Other genotypes may be favored during the summer when populations are dense because these genotypes make for better competitors, the ones more successful at securing limited food resources. Still others may be at an advantage during the lower winter months because they increase the population hardiness or ability to withstand the inclement weather conditions that kill most members of the other genotypes. The fitness of genotypes can also vary according to their relative numbers, and genotype frequencies may change as a consequence. This is known as frequency dependent selection. Particularly interesting is the situation in which genotypic fitnesses are inversely related to their frequencies. In natural population of animals and plants, frequency dependent selection is very common and may contribute importantly to the maintenance of genetic polymorphism. Frequency dependent selection may arise because the environment is heterogeneous and because different genotypes can better exploit different sub-environments. When a genotype is rare, the sub-environments that it exploits better will be relatively abundant. But as the genotype becomes common, its favored sub-environment becomes saturated. So that genotype must then compete for resources in sub-environments that are optimal for other genotypes. It follows then that a mixture of genotypes exploits the environmental resources better than a single genotype. This has been extensively demonstrated. Sexual preferences can also lead to frequencies dependent selection. It has been demonstrated in some insects, birds, mammals, and other organisms that the mates preferred are precisely those that are rare. Dear students, let's now summarize the lecture of gene frequency dynamics. Allele frequency or gene frequency represents the incidence of a gene variant in a population. Alleles are variant forms of a gene or alternative forms of a gene that are located at the same position or genetic locus on a chromosome. In population genetics, gene frequencies refer to the quantity of variation or change at a particular locus or across multiple loci. The gene frequency is different from the genotypic frequency in case of diploid organisms. Although they seem to be related to each other, and gene frequencies are usually calculated from genotypic frequencies. Factors that disturb the natural equilibrium of gene frequencies include mutation, migration or gene flow, random genetic drift, and natural selection. The common conception of evolution focuses on change due to natural selection. Natural selection is certainly an important mechanism of gene frequency or allele frequency change, and it is the only mechanism that generates adaptation of organisms to their environments. Other mechanisms, however, can also change allele frequencies, often in ways that oppose the influence of selection. A nuanced understanding of evolution demands that we consider such mechanisms as genetic drift and gene flow, and that we recognize the errors in assuming that selection will always drive populations towards the most well adapted state. Dear students, it was all about today's lecture regarding mechanism for dynamics in gene frequency or allele frequency. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.